Hello, myself Dhruv and my other groupmate is Somasekhar Reddy Kumala, Lokesh Naik, Rafia Pathan, Hathvi Valera and Vyash SV. Today, we all are going to discuss on development of integumentary system. The skin is the largest organ on the body. So let us know about tissue, organization of the skin, integumentum. It is made up of three layers, the epidermis, dermis and the subcutaneous layer, hypodermis. Basically, epidermis is the outmost layer of skin on our body. It protects our body from harm, keeps our body hydrated, produces new skin cells and contains melanin which determines the color of our skin. Fibrous tissue beneath the epidermis which called dermis. And now third is subcutaneous layer. Subcutaneous layer is the bottom layer of skin in our body. It has many important functions including storing energy, connecting the dermis layer of our skin to our muscles and bones, insulating our body and protecting our body from harm. This is the picture of skin structure. There are given many type of skin parts and skin layers like stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, stratum basals, dermis, hypodermis. Structure of skin route of skin penetration. The main route of transport for water-soluble molecule is transcellular. It involves the passage through the cytoplasm of corneocyte and lipid arrangement of the stratum corneum. The pathway of transport for lipid-soluble molecule is intracellular. It is implicates the passage apparently through the endogenous lipid within the stratum corneum. The transcellular and intracellular route is collectively known as transepidermal route. Now let's talk about germ layer of origin. Any of three primary cell layers formed in the earliest stages of embryonic development, consisting of the endoderm, which is the inner layer, the ectoderm, which is the outer layer, and the mesoderm, which is the middle layer. Epidermis specialized from the surface, ectoderm not involved in the developing nervous system. Dermis differentiate from mesenchymal cells of mesodermal somites, dermatomes. Now look at this figure. It's shown cross section of 24-hour chick embryo in primitive tree area. Let's delightful discussion on this topic. It is 24-hour 4 pair of somite stage of chick embryo. At this stage, the dark peripheral area, opaca and central translucent and colorless area, pellucida are distinctly visible. In the anterior part is present the promonium, which is a small and comparatively more translucent region of area pellucida and is characterized by the absence of mesoderm. In the middle of area pellucida, in this posterior half runs a primitive stick with a primitive groove in the center. The primitive groove is bound of primitive folds. In the anterior half of area pellucida, in the middle runs the neural grooves bound by neural folds. The primitive stick and neural groove are separated by a Hansen node having a small depression in the center, the Hansen spit. Immediately below the primitive groove, the primitive stick gives rise to a small outgrowth, the knot cord and the either side to mesoderms. In the era pellucida, embryonic and extra-embryonic regions also become a distinguished. In the anterior most part of the ectoderm has given rise to head fold, which is a pocket-like extension of neural folds. The underlying endoderm is also transformed into a pocket-like foregut. The promonium is greatly reduced. In front of Hansen node, the mesoderm of the embryonic area differentiated into 3-4 pair of mesodermal somatics. The neural canal is the region to head folds give rise for forebeing. The foregut extends of the either sides into the amino acid vesicle. Now we will be discussing about epidermis. Let's just focus on main point of epidermis and try to remember. First point is embryonic ectoderm, originally a single sheet of cuboidal cells. Second point is developed to become two-layer in the fifth week. Third point is peridermal flattened cell. In outer side surface, cuboidal basal cell, reproductive cell give rise to new layers periderm above them. Fourth point is during 3 or 4, epidermis consists of 3 cell lower periderm. And at last point is intermediary stratum cuboidal basal cells. Now look at this figure. This figure is shown layer of epidermis. Let's discuss about some layer of epidermis. Mesenchyma derives from the middle embryological germ layer. The mesoderm, the outer germ layer known as the ectoderm, also produces a small amount of mesenchyma from a specialized structure called the neural crest. Here we all are able to show peridermal cell. So the periderm is the singular of flattened cell that cover the developing epithelia with a continuous layer. The periderm is believed to persist of the surface of the developing epidermis throughout the embryogenesis until shortly before birth. Here, the figure shows that conferred layer. It is a barrier between the body and the external environment. It prevents the penetration of allergens, pathogens, virus and bacteria, chemical irritants and UV radiation, and the loss of body fluids. It is also mechanical protection and antioxidant barrier. Next figure shown basal layer. It is the innermost layer of the epidermis and contains small round cell called basal cells. The basal cells continuously divided and new cells constantly push older ones up toward the surface of the skin, where they are eventually shed. And at last, we can all see intermediate layer. It typically consists of species such as a malic anhydride grafted polyethylene or copolymer of malic anhydride and polyethylene, which can react with the epoxy inner layer, along with a co-terpolymer compatible with the polyolefin outer layer.
After the fourth month, the epidermis becomes stratified. Epithelia consists multiple layers of cells. Stratum germinative basal cells and their immediate descendants next above. Stratum granulosum layer of cells next above. Stratum germinative consist cells containing keratohylum granules. Stratum lucidum next above. Stratum granulosum thin and clear cells containing degenerative keratohylums. Stratum corneum flat cells on the surface. Cell cytoplasm undergo cornification to become cornified dead cells with degenerate nuclei. The cornification is not intrusive in some areas. Cell layers of lips and annuals. This is picture of epidermis. Emigrant cells invading epidermis. Epidermis consists of cells derived from ectoderm. Proliferation of stratum germinative and foreign cells from outside that migrate to epidermis during embryonic development and remain there in adult. Melanoblast derived from neural crest migrate to dermis and penetrate epidermis in the third month. Melanoblasts differentiate to from pigment granules. Melanosomes to become melanocytes. The pigmentation intensity varies among races, although the number of melanocytes is not significantly different from race to race. The pigmentation is depend on the activity of tyrosinase convert tyrosine to melanin. Langerhans cells morphologically indistinguishable from carotenoids epidermal cells. However, they are recognized by histochemical methods. They have membrane-bound ATPase. They have specific surface antigen different from epidermal cells derived from precursor cells in bone marrow, migrate to epidermis and function as antigen present cells that process antigen entering epidermis. Their antigen is fragmented and presented it to T lymphocytes mediate cellular immune response to antigen. Dermis, skin component underneath the epidermis composed of fibrous connective tissues developed from lateral wall of somites, left and right dermatomes. The mesenchyme of dermatomes from collagen fibers and elastin fibers during the 4-6 months of pregnancy. The deepest layer of dermis, subcutaneous layers, loose and fatty connective tissues contain capillary blood vessels and sensory nerve ending. This picture of cross section of 18-hour chick embryo in smooth hair.
So this is a figure of cross sections of 48 hours chick embryo in somatic area. The positions of embryo with respect to the yolk changes strongly about 48 hours after fertilization. The outgrowth of the cranial features is so strong that the forebeam and hindbeam muscles become almost located to each other. The cephalic regions of the embryo is twisted in such a manner that the left sides come to the lie next to the yolk. The embryo takes now the shape of C. The heads become covered by a double fold. This fold definitely established the first extra embryonic membrane outside the embryo, the meiotic membrane. A few branchioles are grown already visible. The lens placoda will form the lens vesicle. The optic vesicle will become the optic cup and auditory placenta, the auditory pit. The heart differentiated into the four compartments: the sinus venosa connected with the veins, the atrium, the U-shaped ventricle, and the bulb cords. The atrium and ventricle are well distinguishable in this figure. Now, the 10 peak embryo. The study of the facial developments of the peak embryo can be done by tracing the slides along the branchial arc level. Stomatium, which is cephalotherically surrounded by the maxillary prominence and caudal by the medullary prominence. More dorsal in sections is pharyngeal region characterized by the bar-like thickening of the second to fourth bronchial arches. The cleft between the developing eye and the maxillary is the nosococrimal groove. In some places, the epidermis undergo modification and develop into the skin glands and heads. The hair forms epidermis through grow downwards into the dermis to become hair follicle. The mesenchyma at the basal of the follicle form hair papillae surrounded by the hair follicle root sheath. The cells in the gremial matrix sweat glands located throughout the body originated as a downgrowth from the epidermis into the underlying mesenchyma dermis. As it elongates, its ends coils to form the primordium or secretory part of the gland. Glands become to function shortly after birth. Large procaine sweat glands are mostly confined to the axial pubic, perineal regions and the areolae of the nipples. These glands open into the upper part of the hair follicles. The mammary glands, specialized sweat glands that develop as a solid epidermal downgrowth into the underlying mesenchyme, develop from thick strips of ectoderm extending from axillary to inguinal region, mammary ridge. Each primary bud gives rise to several secondary mammary buds that develop into lactiferous duct. Canalization of this bud is induced by placental sex hormones entering the fetal growth circulation. The gland is supported by fibrous connective tissue and fat that develop from the surrounding mesenchyme. Tissue cell culture of skin. The epithelial tissue cell of the skin are potentially growth in vitro and prepared for various medical purposes, advantageous for the replacement of skin damages, burn, wounds, etc. The tissue graft are harvested from undamaged area of donor site. Autograft, isograft, allograft, xenograft. Skin allograft or xenograft can be used as a temporary dressing rejected by the body immune system in a short time. Autograft and isograft are more acceptable, recognized as self-component by the body of immune system. Epithelial graft may be obtained by growing keratinocytes of the laboratory. The graft is cultured from a small piece of patient own skin. Grow enough epithelial graft to cover 10 into 10 centimeter damage within 3 weeks. The individual graft are typically multi-layer cell much like epidermis. As the graft is placed on the acceptor wounded tissue, the cell become increasingly differentiated. Basal layer, germinal layer, top layer takes on the protective role of the skin by becoming conifer lining of the surface.
Anomalies and genetic diseases of the skin, congenital failure of normal differentiation. Skin retain its fetal character. failure of pigment production, deposition in the epidermis, albinism, overabundant pigmentation production, melanism. A typical pigmentation give pigmented spot mole, rough scaly skin due to abnormal conification of superficial layers. Ichthyosis show thick plates of epidermis. Acquired diseases abnormality, zero dermapigmentation, XP, hypersensitivity to UV light and high incidence of UV induced skin cancer. Mutation leads to lack of enzyme necessary for excision, repair of DNA. UV light characteristically cause thymine dimers. The dimer should be excised by exonuclease, leaving a portion of damaged DNA. Genetic disease of the skin, inherited from one generation to future generation. The inheritance may be dominant or recessive hereditary fashion. Keratosis filariasis, dry and worst pattern of skin, chicken skin, inherited of autosomal dominant fashion. Affected parent may inherits the disease to all of or 50% of his her children. KP may also manifest in patients due to deletion of chromosome 18, translocation of 18p chromosome to Y chromosome, involve LAMA1 gene situated in 18p locus, express protein coded 29 of 32b mRNA, epidermolysis bullosa EB, rare genetic abnormality, 2 persons per 1 lakh people in USA. Abortion of skin due to abnormality of lamina 5 production structure, lead to destruction of collagen 8 at the upper layer of dermis, failure to maintain epidermis stabilization, inherited in all autosomal recessive fashion, transmitted by healthy carrier of both parents. Thank you.